this is the second part of our third part of our chapter, which is body fluids in circulation. So till now we have covered till the formed elements, and we have discussed about leukocytes a bit, and so we are going to start up with the leukocytes again. So what are leukocytes? Leukocytes are nothing but are leukocytes are nothing but are WBCs only. WBCs have another name which is leukocytes, and we have talked about RBCs have another name which is what? Platelets have thrombocytes. Okay, and we have erythrocytes, and the leukocytes are the RBCs only, and we are going to discuss that they are colorless. They don't have color, red color or any other color due to the lack of any pigment, and due to the lack of hemoglobin they look colorless. Okay, they are nucleated, whereas RBCs were devoid of nucleus. Okay, and they are relatively lesser in number, and it is about 6,000 to 8,000. Okay, 6,000 to 8,000. Okay, and is about 6,000 to 8,000. Leukocytes are generally short-lived, whereas RBCs have the average lifespan of 120 days, and thereafter it goes to get destroyed in the spleen. Remember, and the leukocytes are generally short-lived, and we have RBCs of two kinds, which are granulocyte, which will look like grain-like structure, we can say, or agranulocytes. Okay, granulocyte which have the grain-like structure, agranulocyte which lacks that structure, and granulocytes are further divided into three parts, and agranulocyte is divided into two parts. What are these parts? Granulocytes have two parts, which are neutrophils, eosinophils, and basophils. Neutrophils are neutral, eosinophils are acidic, whereas basophils are basic. So from the name it's pretty much clear to us that how they function. Like neutral will be neutral, eosinophils will be acidic, and basophil will be basic. And these are the granulocytes type, and agranulocytes have two types, which are lymphocytes and monocytes. If we are talking about the abundancy, the neutrophils are the most abundant, which is about 60 to 65% of the total RBCs. And if we are talking about basophils are the least abundant. These are the least abundant. It is about 0.5 to 1% only. And neutrophils and monocytes are what? These are phagocytic. What are phagocytic? Phagocytic are those cells which used to destroy the foreign material. Like this is a foreign material, when it will enter the cell it will be get destroyed, because we know our monocytes and neutrophils know that they are foreign material and they can harm our body, or they can harm our cells, so they used to destroy them. Okay, so this is the role of them. And next it is written that yes, basophils secrete some of the hormones like histamine. They secrete histamine, serotonin, and heparin. These are secreted by what? Basophils. These are secreted by basophils, and these are involved in the inflammatory reactions. Eosinophils is 2 to 3% will resist the infections. They will resist infection, and this is how they look. Like RBCs look like this. It looks biconcave in shape, right? And there's a platelet. Eosinophils, you can learn it by that it is acidic, so it will look like red in color. It is basic, so it will look like somewhere dark blue or a little bit of purple, and it will look like pink. Okay, you can learn it by this. And here it is written that eosinophils resist infection, and they are associated in the allergic reactions also, associated with the allergic reactions also. And lymphocytes, which is 20 to 25%, are divided into two kinds also. So lymphocytes we have two kinds, B and T lymphocyte. Okay, and both are responsible for the response of our body, immune response of our body. So they also they works in the defense mechanism of our body, defense mechanism of our body. Okay, and next it is written that I've already told you that platelets are the thrombocytes, or are also thrombocytes are commonly called as platelets, and these are cell fragments which are produced by megakaryocytes. What are megakaryocytes? These are the special cells which is present in the bone marrow, and megakaryocytes are from the megakaryocytes our platelets gets produced. Okay, and it is written that blood normally contains this amount of platelet, like 1 lakh 50,000 to 3 lakh 50,000 of platelet. Everyone, so let's continue. And we were been talking about the RH grouping. So I need to talk about that also, that in the platelets, which we call as thrombocytes, the thrombocytes, which we commonly call as platelets, are produced by the megakaryocytes, and the number of platelets is written here. And platelets can release a variety of substances which are involved in the coagulation. We know that the main function of our what platelets is to coagulate or the clotting of our body blood, and the reduction in the number can lead to some clotting disorder. I know one name of clotting disorder which is called hemophilia. And due to the reduction in the number, there can be excessive loss of water blood from the body. Okay, so next we are going to talk about the blood grouping. So we have two kinds of blood grouping. First one is ABO grouping, and second one is RH grouping. So what is ABO grouping? ABO grouping is based on the presence or absence of two surface antigens, whether the antigens are present or not. Okay, so whether the substance antigens are present or not. Okay, and we on the RBC, namely A and B. Similarly, the plasma of different individual contain the two natural antibodies. Okay, and the distribution. Okay, we have the four groups of blood: A, B, AB, and O. Okay, so this is what our ABO grouping is. If the antigen is present or not, it tells our ABO grouping. For example, it will become more clear to you from this table. Like we have A blood group, B, AB, and O. We have four blood groups in total. Antigen will be same as the blood group. Like A will have the A, B will have the B, AB will have A and B, and O will have nil. And antibodies will be different from the antigen. Like if it is A, the antibody will be anti B. For B, it will be anti A. For AB, it will be nil, and for nil, it will be anti A and B. And the donor can be for antigen A, donor can be A and O. For antigen B, donor can be B and O. For antigen AB, donor can be AB, A, B, and O. And for the antigen O, the donor can be only O only. And from here you can notice something, that here, in the in all the blood group there is O, means that O is the universal donor, as it can donate to everyone. So here it shows that O is the universal donor, and we also have a universal recipient, which is receptor. What is it? It is AB. It can take it from any blood group, and O can only take it from O O blood group individual, and therefore it is called as universal donor because it can donate to every individual having any blood group. Okay, so and next one is RH grouping. So what happens in the RH grouping? It is in another antigen, and it is similar to the one which is present in the monkey. Okay, and this also observed on the surface of RBCs, which is 80% of humans. Individuals which have the presence of this antigen are called as RH positive, and the antigen which in which it is absent is called as RH negative. RH positive and RH negative. If a person Look, if a RH negative person is exposed to have the blood of RH positive blood, then it will have the specific antibodies against the RH antigen. So RH group should be matched before transfusion. So it is already it also, while doing the transfusions, we need to check whether the RH group is matched or not. Okay, so in case of RH incompatibility, like this matching, if the RH is not matching, then what happen? Then we can